sorry about that. Chapter 15, continued. You can't assort independently if you're located on the same chromosome. Genetic recombination is a term that we use to refer to the production of offspring with combinations of traits differing from either parent. In your parental types, your offspring will have a phenotype that matches one of the parental phenotypes. Recombinant types, these are recombinant offspring or recombinants. These are offspring that have non-parental phenotypes. In other words, they have new combinations of traits. There is a 50% frequency of recombination if we compare any two genes on different chromosomes. Another important thing during meiosis that causes recombinants is crossing over. Remember, crossing over is the exchange of equivalent DNA between homologous chromosomes. This is an important creator of variation. Variation is important for natural selection because recombinant chromosomes plus random fertilization gives us a lot of raw material, a lot of new combinations of genes that allow for natural selection and evolution to occur. Sturtevant was one of the first people to create what is called a genetic map. And a genetic map is a list in order of the genetic loci, that is the plural for locus, which means place, along a particular chromosome. And he predicted the further apart that two genes are, the higher probability there is that crossing over will occur and that you will get a higher recombination frequency. A linkage map is a genetic map of a chromosome based on recombination frequencies. When we're doing a linkage map, we use the term map units. Map units are derived from the distances between genes, and one map unit equals 1% recombinant frequency. So remember, these map units are going to indicate relative distance apart, relative order, and not precise locations of genes. Okay. Changes in chromosome number and structure, particularly in the development of an embryo and fetus, can lead to miscarriage, also known as spontaneous abortion, or some fairly dramatic developmental disorders. Plants, it's very interesting, tolerate these genetic changes much better than animals. So if you'll take a look at figure 15.13, it shows you what happens when this non-disjunction occurs. This is when pairs of homologous chromosomes don't separate normally during meiosis, and this gives your gametes the wrong number of chromosomes. Some of the gametes have too many and some have too few. Aneuploidy is the term that we use that is the result of fertilization between gametes that have already experienced non-disjunction. And what's going to happen is when the gametes that have the wrong number of chromosomes join one another, it's going to give offspring with an abnormal number of a particular chromosome. And the effect on the offspring will depend on what genes and what chromosome the problem occurs with. A monosomic zygote has one copy of a particular chromosome instead of two. A trisomic zygote has 3 copies of a particular chromosome. Polyploidy is the term that we use when an organism has more than 2 complete sets of chromosomes. If the organism has triploidy, then they are what we call 3N. They have 3 sets of chromosomes. Tetraploidy, the organism is 4N. They have 4 sets of chromosomes. Polyploidy is very common in plants because for some reason they seem genetically to be able to handle the abnormalities in chromosome number much better than animals. Polyploids are more normal in appearance, which is very interesting, than aneuploids. Chromosome breakage can also alter chromosome structure. If you have a deletion that actually removes or deletes a chromosomal fragment. If you have a chromosomal duplication, then you get a segment that is repeated. An inversion simply flip-flops a segment within a chromosome. It reverses its orientation. Translocation actually moves a segment from one chromosome to another. All of these have varying effects on the phenotype. See figure 15.14. It will show you some of these aspects. So take the time to study figure 15.14, and then I'd like for you to look at 15.15. The term that we use for the set of symptoms that arise because of changes in your genetics is syndrome. For example, Down syndrome is an aneuploidy condition where the person has 3 of chromosome number 21. And interestingly enough, the odds of this happening increase with the age of mom whenever she gets pregnant. With aneuploidy of sex chromosomes, remember, non-disjunction is responsible for the aneuploid conditions. And many of these we have talked about. Remember, Klinefelter, an extra chromosome in the male. For example, XXY, XXX for the female. And typically, they're healthy. There's no unusual physical features, but they are at risk for learning disabilities. With XXX, they're healthy. It is the only known viable monosomy in humans. Let's talk about disorders with structurally altered chromosomes. These were also in your PowerPoint that we did when we were doing meiosis. Cri-du-chat is when you have a deletion in chromosome number 5. It gives the child a very distinctive cat-like cry. And this child is intellectually disabled and usually dies early in infancy or early childhood. Translocations of chromosomes can cause certain cancers like chronic myelogenous leukemia. See figure